When we think of passing along genes, we typically think in terms of passing genes to offspring. That process is termed vertical gene transmission, from parent to progeny. However, the microbial world, especially bacteria, contain members that are especially adept at what is termed horizontal gene transfer. That is, the cells can pass along usable genetic information to other cells that are not progeny cells, but are neighbors. Horizontal gene transfer occurs via a few different mechanisms termed transformation, transduction, and conjugation. Each of these mechanisms will be discussed separately. Here we discuss transformation. The phenomena of transformation was first observed by a researcher working with pathogenic and non-pathogenic strains of Streptococcus pneumoniae. The pathogenic strains grew as smooth-looking colonies on a petri dish and were named the S strain. The non-pathogenic strain was named the R strain for the more rough-appearing colonies on the same medium. The famous Griffith experiment is depicted in the figure from your text. In the first panel, you can see the results of what would become the known control conditions. Live S strains injected into mice caused illness and death of the mice. Heat-killed S strains introduced similarly did not kill illness and death, indicating that live organism and not some toxic component was responsible for the effect. The R strain did not kill mice and thus has lost something important in the disease-causing realm. The key experiment is shown in the middle panel. Here, when Griffith injected a mix of living R cells and heat-killed S cells, the mice died. Surprisingly, when he re-isolated the bacteria from the diseased mice, he found live S cells. What happened? Somehow the R strains were transformed into S strains. He could not know why, since the genetic material had not been defined. Later, it would be shown, as depicted in the last panel, that DNA was responsible. The R strain had a mutation in the capsule gene which prevented it from making a capsule, and thus was not pathogenic. In the mixture, the R strain took up the DNA of the dead S strain and recombined. By doing so, it was able to restore a functional capsule gene, became pathogenic, killed the mice, and grew a smooth-looking colony on the petri plate of medium. Hence, the uptake and recombination of DNA by a cell is known as transformation. The next type of horizontal gene transfer involves bacteriophage, which are viruses that infect bacteria. Bacteriophage will be discussed in more detail in another chapter, but let's get a quick overview here. Some phage only undergo the lytic cycle, which is shown on the left. The phage infect the bacteria, replicate, assemble new phage particles, and lyse out to infect new cells. In some phage, called temperate phage, a lysogenic cycle can occur. In this phase, depicted on the right, the virus inserts into the chromosome. It will remain there and replicate with the bacterial divisions. Occasionally, it will re-emerge from the chromosome and enter the lytic cycle. As mentioned, transduction is a horizontal gene transfer that involves bacteriophage. There are two types of transduction, generalized and specialized. Each utilizes a separate path of the bacteriophage infection, lytic or lysogenic. We will discuss the generalized first. In generalized transduction, it's important to note that any gene can be transferred from one bacteria to the next. This is because of the viral infection process involved. As depicted in the figure, during a lytic infection, the virus infects the cells and begins to replicate. In the process, the bacterial chromosome is degraded and broken up into pieces. If one of the chromosomal pieces is the right size, it can be packaged into a phage particle. The piece of packaged DNA can be from anywhere in the chromosome, and the transducing particle will not have viral DNA, only bacterial DNA. When the particle is released by the previously infected cell, it can attach to a new cell and inject the bacterial DNA. This can be incorporated into the new host, making it a recombinant cell.